Hello students, in today's class we'll be studying about types of phytohormones or plant hormones. So there are 5 types of plant hormones or phytohormones which we'll be studying. First 3 of them are considered to be plant regulators, and the next 2 are considered to be the plant inhibiting hormone, the growth inhibiting hormone. First 3 are the growth regulators and the next 2 are the growth inhibiting hormones. So firstly we'll talk about the plant growth hormones, which are auxin, gibberellin and cytokinins, and the inhibiting are ethylene and abscisic. So we'll start with auxins. When we are talking about the phytohormones, the 2 things which comes in our mind is the site of synthesis, where these hormones are synthesized, and what is the function of it. So what is the site of synthesis of auxin? It is in the shoot apical meristem and the young leaves. So auxins are synthesized in the shoot apical meristem and the young leaves. Their first function is that it promotes the cell elongation. It promotes cell elongation. It helps in elongation of stem, as well as it suppresses the growth of lateral bud by apical bud. Now this is the major function of auxin, for which auxin is known. So the auxin is known for apical dominance. This entire process is known as apical dominance. What is apical dominance? The apical bud dominates over the lateral buds. The process in which the apical bud dominates over the lateral buds. When the apical bud grows, then the lateral bud formation will be very minute, will be suppressed, or you can say. So the apical bud suppresses the growth of lateral buds, and hence the apical bud dominates. This process is known as apical dominance. Now we move on to another function of auxin, which is the growth of parthenocarpic fruit. Auxins helps in the growth of parthenocarpic fruit, means the seedless fruit. It helps in the growth of seedless fruit like banana. Now, auxins also control the phototropic behavior. Now, the question comes, give reason how these auxins are controlling the phototropic behavior of the plant. Now, for this, we'll see this diagram. Here, the plant stem is moving towards the light, and hence it is showing the positive phototropic behavior. Now the question arises that this positive phototropic behavior is shown by the plant stem. How does this hormone is contributing in this behavior? So how does this auxin is contributing in this behavior? So your answer is here. As the plant stem receives the sunlight, the auxin molecules are produced in that region, and slowly and steadily these auxin molecules start moving towards the region where the sunlight is coming the least, and where in whichever region, that means the shady region, where they reach, actually the auxins reach the region which is receiving less of the light, the shady region, and that region the cell elongation will take place maximum as the auxin has accumulated in that region more. Now, due to the cell elongation more in this region, it will lead to bending of the stem towards the light, and hence this auxin molecule, this auxin hormone is helping in the stem showing positive phototropic behavior. This auxin hormone helps in the stem showing positive phototropic behavior. Now, we move on to another function of auxin, which is that it controls the geotropic behavior of the plant as well. Now see, this diagram in which the stem is showing negative geotropic behavior. Geotropic means the movement towards the gravity, the growth movement of the plant towards the gravity or away from the gravity. So here we can see that the shoot or the stem shows negative geotropic behavior. It moves away from the gravity. So this negative geotropic behavior is also because of the auxin molecule. How? Auxin gets accumulated more over this region. So due to the excessive accumulation of auxin in this region, the cell elongation takes place maximum and leads to bending of the stem, and hence the stem moves away from that of gravity and shows negative geotropic behavior, which is due to this auxin hormone. Now we move on to another plant regulating hormone, plant growth regulating hormone, which is gibberellin. It is produced in the meristem of apical buds and roots. Its first function is that it promotes the cell elongation in the stem region. Next function is that it breaks the dormancy of the seeds. Now this is the major function of gibberellin, for which it is known. It is known as the hormone which helps in seed germination, and the hormone which breaks the dormancy. What is dormancy? The inactive period in which the seed remains inactive and doesn't show any growth. So it breaks that inactive period and helps in seed germination. Another function of gibberellin is it delays the senescence. Senescence means the process in which the leaves start showing yellow. It is a process of aging in which this leaf starts becoming yellow and starts falling off. So it delays the senescence, it delays the aging and hence regulates the plant growth. Another function of gibberellin is it induces the parthenocarpy, it induces the formation of parthenocarpic fruit. Then the last function of gibberellin is that it causes foolish seedling disease. The symptoms of foolish seedling disease is that in this case the shoot region of the plant grows to the maximum height. It receives maximum height and the leaves become pale, and it was observed that this happens because of the fungus known as Gibberella species. So this Gibberella species was responsible for causing the foolish seedling disease, and this Gibberella is the fungus. Now, when the scientists have found the reason behind the excessive growth, they found that it was because of this excessive gibberellic acid in that particular region of the stem, that this excessive stem elongation has occurred. So, they have concluded that gibberellin is responsible for stem elongation. So the gibberellins was first, you can say, was first found in this, the major function of gibberellin was first being found in this rice plant in which the foolish seedling disease occurred because of the fungus gibberella. Now the next hormone is cytokinins. Cytokinins is found in the roots. It is synthesized in the roots first and then it moves to other parts of the plants. So it stimulates, the major function of it is that it stimulates the cell division and helps in enlargement of fruits and seeds. It helps in enlargement of fruits and seeds. It also breaks the dormancy of seeds and breaks the inactive period of the seeds and helps in the seed growth, the growth of the seedling. Now we move on to the growth inhibiting hormones. We have now moved on to the growth inhibiting hormones. There are two growth inhibiting hormones, ethylene and abscisic acid. Ethylene is also known as gaseous hormone. Ethylene is also known as a gaseous hormone. It is found in the gaseous state. In senescent leaves it is found, and flowers, germinating seeds, and ripened fruits. This ethylene hormone is synthesized. Now, the function of ethylene, for which it is also known as the hormone which is responsible for fruit ripening. For example, if we take a green papaya and cover it with a newspaper and leave it fortnight, and then we see it in the morning and remove that newspaper, we'll see that we get a ripened papaya. Why this happens? This happens because of the ink present in the newspaper contains ethylene, and ethylene is the hormone responsible for a ripening of the fruit. When this ethylene comes in contact with the green papaya, it helps in ripening of it, and hence we get a ripened papaya in the morning. So we can say ethylene is the fruit ripening hormone. Ethylene also promotes senescence. Now see, this is completely opposite. This is the complete opposite function of that of gibberellin. Since gibberellin was a plant growth regulating hormone, it used to delay the senescence. But ethylene is the plant inhibiting hormone, hence it is promoting the senescence. It is promoting the aging process, the senescence process. It also stimulates the abscission layer formation. Now, what is this abscission process? Abscission is a process of a separation. When the leaves fall, then they leave certain mark over that region. When the leaves fall, then they leave a mark over the shoot region, and that mark is known as the abscission layer. The point of separation of the leaf from the shoot leaves a mark which is known as the abscission layer, and the process of separation of the parts of the plant from the shoot is known as abscission. So the process of separation is known as abscission. So this ethylene hormone helps in the formation of abscission layer. Now, another function of ethylene is that it causes epinasty. Now, the question arises, what is epinasty? So, epinasty is that process in which when the leaf upper portion becomes, it gets the excessive growth, becomes bulky enough. When the leaves upper portion gets excessive growth as compared to the lower portion, then it leads to bending of the leaf. This process is known as epinasty. Now we move on to another plant growth inhibiting hormone known as abscisic acid. Abscisic acid is also known as the stress hormone, and the site of synthesis of abscisic acid is in the fruit and in mature green leaves. It also inhibits, the function of abscisic acid is that it inhibits the seed germination. It inhibits the seed germination and hence is known as the plant inhibiting hormone, plant growth inhibiting hormone. It stops the seed germination. It induces dormancy. It helps in the dormancy state, in getting the inactive state. Hence, it is stopping the plant growth. It stimulates the closure of stomata. Now, this question answer we are getting over here, why abscisic acid is known as the stress hormone. It is known as the stress hormone because during the stress conditions like excessive heat, what happens that these excessive abscisic acid is released in excessive amount during the adverse condition, during the condition of stress. When there is excessive heat, excess of abscisic acid gets accumulated and it results in the closing of stomata, Stomata then closes and prevents the loss of water from the leaf. So it helps us in preventing the loss of water with the help of abscisic acid by the closure of stomata. Now, the another function of abscisic acid is that it promotes the wilting of leaves. Wilting means decaying of leaves. It helps in decaying of leaves. So we end up with this topic of types of phytohormones, as well as we end up this chapter here. Thank you, class.